Kia ora. Welcome to Science with Folly. Today we're going to be learning about DNA mutations, and in particular a certain kind of mutations which we call point mutations. So here we have some genetic code. As you can see, the top line has got T's in it. So what is it? It's DNA. Down here, this line has got U's in it, so it's RNA. So we've got DNA, which is transcribed to messenger RNA, and then that messenger RNA gets translated to produce these amino acids down the bottom here. You should all be familiar with this table here. It's the codon table, which shows which codons code for which amino acids. So if you looked on the codon table, you would see the AUG codes for MET, which is the start codon. UGG codes for TRP, and so on, and so on, and so on. These are all amino acids. UGA, does that code for amino acid? No, it codes for stop. Now, mutations in the genetic code, they can occur due to copying errors during DNA replication. And the first kind of mutation we're going to look at is called a substitution mutation. So at the top here, we've got our original coding sequence. But down here, you can see that C has been substituted with T, which means it gets transcribed to an A in messenger RNA. Now this changes the codon. So we still start off with AUG, which codes for MET, but then we've got UAG, which now codes for stop. And as you can see, that would be our sequence there. We would have a start codon and then a stop. This is called a nonsense mutation because the polypeptide chain is going to be stopped early and will not function at all. The next type of mutation we're looking at is called an insertion mutation. And in this case, a base gets inserted during the replication of DNA. Here we have a G gets inserted between the C and the A, and obviously that gets translated to a C. And what happens is that the first codon remains the same in this case, but subsequent codons get moved across. And you can see that now we have MET to start off with, but CUG now codes for LEU, and we get a whole different amino acid sequence. This is called a frame shift because all the codons are moved across. This kind of mutation is also called a missense mutation because you still get a polypeptide chain, but it's different to what you would have had produced from the original. The last type of mutation we're looking at is called a deletion mutation. And in this case, during replication, a base gets deleted. And now instead of having G, G, C, G, the C has been deleted and it goes G, 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 which means we have another frame shift because what happens here, you can see the codons get all changed after that deletion. And we end up with a different polypeptide chain after that deletion. As you can see, the first three amino acids are the same as the original, but this one and this one have changed. And so therefore, a polypeptide chain is still produced, but it's different, and therefore it's a missense mutation. Here's another kind of substitution mutation I'd like you to have a look at. In this case, what has happened is the G up here has been replaced by an A, which transcribes results in a U. And so our codon changes to AAU instead of AAC. But as you'll notice, they still code for the same amino acid. And this is due to the redundancy of the genetic code, which is an important thing you should know about, shouldn't you? And in this case, it's a substitution mutation, and we call it a same sense mutation because it still gives us the same amino acid. So just to revise some key words you should have picked up, we've got substitution, deletion, and insertion, and that's what happens to the bases. And then same sense, missense, and nonsense, and that relates to the amino acids that are produced. So thanks again for watching. Hope you've learned something or at least revised something. And don't forget to subscribe.